Could I mention the Portland meeting was very nice. I'm sorry to see there weren't very many Seattle people down there. Sally Newbar came down from Bellingham.
Hey, listen, there was a husky
junkie. Well, I know. But I mean, still, it was a really very well done meeting.
So I've got no disclosures to make. I've got several papers to go over. This particular one about insulin allergy was in diabetic medicine. It's really meant for non-allergists, so some of the concepts are fairly superficial. But I had a patient the other day that was referred to me for insulin allergy, and that required me to kind of delve into the literature to remind myself what that was all about. We haven't talked about it much, so I thought it might be interesting to go over some of this stuff. This was a bigger problem when we used animal origin for the insulins, and has decreased significantly with the recombinant human insulins. Paper quoted an estimate of 0.1 to 3% of patients treated with insulin have evidence of insulin allergy. I think based on the frequency that I get patients with that problem, it's got to be on the low end of that. Another study looking at patients referred for the issue of insulin allergy, about 60% of them have some other cause for whatever symptom they're having. So it's not all that common. Primary antigen tends to be insulin, but there's some additives in the various insulin products that have been reported as the cause for the clinical symptoms as well. Latex is part of some of the syringes used and has at least been reported in a few cases. The recombinant product has potentially some yeast or bacteria products left over, and that's been speculated as potential cause for reactions as well, but it didn't really present any cases, so I don't know whether anybody's ever actually chased it down to the point where that's actually the antigen of concern. One of the things I had to do was look up the various insulins, remind myself what the names are, what additives they have, and this chart kind of summarizes that from several different sources. Most common allergic reactions are the IgE ones, and most of these tend to be just local reactions. Less commonly are going to be urticarial angioedema reactions, and fairly infrequent issues are anaphylaxis. Other types of immune responses with immune complex reactions and delayed hypersensitivity can happen as well. So this particular paper centered itself around several cases that they reported that provided some variation in the problem and the decision making that went on. I'll just kind of review each of these. 65-year-old gentleman with a pancreatectomy, which was the reason for his need for insulin. He started developing local skin reactions, red, indurated, and itchy. He was initially on Humulin NPH, and they switched to Novodisc NPH, and that didn't change anything. So at that point, they started evaluating him further. He had skin tests that showed ID reactions to essentially all the insulins that they tested. They chose Detemir and Lispro as the sort of best alternatives of the antigens or the insulins that they had available, and put him on antihistamine. And this functionally solved his problems. He was able, gradually over the course of a few months, his local reactions did go away, and his follow-up for years had no further problems. I think that shows up fairly well. The decision here was based on kind of sketchy differences between the size of those skin tests, that they're being kind of the smallest one. But that seems to be the conclusion in a lot of these case report series, that people react to multiple different antigens, and you sort of pick the best you can. And that second case was a 40-year-old woman who was hospitalized with ketoacidosis. She was stabilized on IV insulin after things had been normalized again. She was switched to subcutaneous insulin. She developed generalized hives, so they switched brands of insulin, and she did okay initially, but about 4 months later was admitted with anaphylaxis. Again, stabilized her with IV insulin. I'll comment about that in a minute. Then underwent rapid desensitization using subcutaneous aspart. She was functionally switched to a continuous subcutaneous infusion with a pump after that was accomplished. And she generally did well over the subsequent 3 years, occasional hives, but nothing else.
Was that new onset diabetes at that age?
Yeah. That was what was implied anyway.
Yeah.
Didn't really comment about whether she had had problems before in previous instances. And the third case was type 2 diabetes who had been controlled on oral medicines for 19 years, then had NPH started. After about 6 months, he had a regular insulin added, and about 3 weeks after this, had generalized urticaria, which responded to just oral antihistamines. Within a couple of days, he had another episode of hives with angioedema and shortness of breath, and at that point, they skin tested him and got something similar to that previous skin test example, multiple positives, although he did have a few insulins he didn't react to. They chose at that time not to go with those insulins, but to stop insulin altogether, rely on the oral medicines, and change in diet and exercise to control it. But these three cases kind of illustrate the variety of kind of decision making based on difficulty to manage their diabetes and the degree of response problems that they've got. They presented in the paper then sort of an outline of how to approach these problems. I think it's probably more oriented towards non-allergists than allergists, and we would probably reorder these a bit. But at least for mild reactions, the response, if there's some evidence of latex allergy, would be to just change the needles being used. More commonly, it's going to be the insulin, so switch to another insulin is appropriate in management of local reaction with antihistamine. It would be appropriate. With the more severe cases, that makes sense to then do some form of IgE type analysis, and the skin testing is convenient. You can get IgE for insulin as a RAS test as well. And then the response depends on the clinical problem there. If it's type 2 diabetes, you may just be able to stop the insulin and optimize their care in other ways. But then if they require the insulin, you've got a variety of tools to use. IV insulin, subcutaneous desensitization that was done in that case too, infusion pumps, and then a variety of medicines. There are 2 case reports with each of these, including omalizumab as a way of getting patient desensitized. So desensitization, incremental doses leading to development of tolerance, and when you do this, you get mast cell reactivity decreased. This next comment is one that really references the next paper, but just wanted to make it. In this paper, they used the research that said that the internalization of this Fc IgE antigen receptor, the internalization of it into the mast cell was diminished. Next paper, we'll talk about those details. The interesting thing was, in a lot of these case reports, is that IV insulin seems tolerated in a lot of these people with hypersensitivity that is triggered by the subcutaneous mechanism, not really understood. This is their proposed desensitization. It's intradermal and subcutaneous, and it mimics the kind of protocol we use for antibiotics or biologics when you're trying to desensitize somebody. Initial dose is about 1/1000th of a unit, and then it's functionally doubling doses up to a unit and then adjusting the dose necessary for the patient after that. 20 to 30 minute increments between them, so it take you 5 or 6 hours to run through that, assuming no problems. Most of the reports of all of this are all case series, and they use different modalities, which usually is the reason to write the paper, probably. This rapid subcutaneous desensitization, others have used an infusion pump and incremented the dose that way. The interesting reports have been the fact that IV seems to be better tolerated in these patients, and at least one of the case reports was a couple of twin boys who had diabetes, type 1 diabetes. They needed insulin. They started having systemic reactions to their usual injections. They were able to be stabilized with the IV infusion, and they tried the subcutaneous injections, and they reacted to that. So they placed them on IV infusion and then used the subcutaneous infusion and very slowly ramped up the dose. And as they ramped up the subcutaneous infusion, of course, they decreased the IV dose to manage the glucose. And it took them about 140 hours for both of those people. Yeah.
You mentioned that the IV is better tolerated. What's the actual difference between the IV insulin and the subcutaneous?
It's the same stuff.
So they don't change the product at all? They
No.
just change how it's
I mean, it's
delivered?
one of the regular insulins. But,
You know, the people with type 2 diabetes have tons of insulin circulating around. They're not reacting to their own insulin. That's,
the insulins that are used have some configuration
Well, that's
change that makes them the allergen, but what's the thing that's not really well explained, it's just an observation that people have made repeatedly, is that they seem to be able to tolerate the IV, which is fortunate, because that lets us...
Well, that's just it. It does suggest something about the
IV
skin.
is regular insulin.
Yeah.
Whereas all these other ones are modified.
Right.
I mean, you can use regular insulin in the
Yeah, but
subcutaneous.
they're
In the
reacting
pump,
to
do they
regular
use
insulin.
They're reacting
insulin in
to
the
regular
pumps, or is
as well.
that-
See,
Not
there's-
the subcutaneous pump, they use aspirin.
I think
So there's
it
something
suggests
different
something
about
about the
it, too.
skin route. It
Right.
increases the risk.
Well, I would think that I would wonder if anybody's looked, but is it reacting with something in the skin that's causing a
Well,
flare?
I mean, those are the thought games people say that nobody's...
Figured
Is
it
there a difference in frequency between people with type 1 and type 2, since they're quite different diseases? Type 1, more an immunologic disease in the first place. I'm just wondering if the prevalence of these are different in those two conditions.
Didn't find anybody really
The two
providing any
cases
good
you have.
prevalence data for this at all. So.
Look, I mean, that's why I asked about that one patient with a type 1. She's the only one who clearly was reacting to plain old insulin.
Yeah.
And with a true anaphylactic reaction. And that would make
Well,
sense.
the other guy with the pancreatectomy had skin test reactions to the regular insulin.
Right, yeah. But again, I wonder if it's reacting in the skin or something. If it's causing a foreign. So,
While we've interrupted you, just as we have 3 speakers, we're going
Right.
to have to truncate that next
All right. So
presentation.
this addresses that one comment about what happens when you do a rapid desensitization. That article, that diabetes article referred to, was also commented on in this one, and they didn't think the explanation for the desensitization is correct. What they developed was a mouse model with both in vivo and in vitro parts to it. What they did was they passively sensitized these mice mast cells with IgE directed at DNP and ovalbumin. Then they would challenge them with those two antigens, and they could get a reproducible hypothermia in these mice. It was dose responsive and non-lethal. And then they would do a rapid desensitization, incremental doses. And we can talk about some of the details of that if it seems necessary, but this is an example of their protocol. We look at the curves with myself a pointer here. Yeah. Okay. So this hypothermic response is what happens when you just give the dose, that 1.5 milligram per kilogram dose, and it's dose responsive. And then the next is if you follow the red dots, if you desensitize them with incremental dosing every 30 minutes, and then challenge them, you don't get that hypothermic response, and that's highly reproducible. If you do the same process with ovalbumin, you get the same thing, and it's antigen specific. So if you desensitize them, the red dots here, to the DNP, and then challenge them, you've blunted that hypothermic response. If you desensitize them to ovalbumin and give them DNP, it doesn't prevent it. Then they just reverse that process. This is the in vivo model. They harvest the mast cells, and under the different conditions of this, not desensitized, desensitized. Mast cells are stable, their granules are intact. If you challenge them, that initial large dose, they degranulate. They were able to demonstrate that. If you desensitize them and challenge them, they don't degranulate. It's what you'd like to see. This MCPT is one of the chemicals that monitor as part of desensitization, the mast cell degranulation. It's only in the condition of sensitized challenge that gets released. All the others don't have any of that.
I'll go to the conclusion.
Okay. So they have a in vitro model that basically says the same thing. You can see just looking at the graphs that there's only one condition where a response. And this is kind of a graphic example of what they've tagged each of these antibodies. They're predominantly on the surface of the mast cell in the desensitized state, and all of these complexes in the antigens move internal to the mast cell during both the challenge as well as the desensitization. Those cells were out of the in vivo model, but they reproduced basically the same thing with the in vitro model. So, their final conclusion is that internalization of the IgE receptor is the primary mechanism for this, the rapid desensitization and preventing degranulation.
All right, looking at, I'll have to leave it there and move on to the next speaker. Anybody else seeing? I haven't seen a patient with insulin allergy in years.
In local hives with injection, but all we had to do was put them on antihistamine. That's it.
Yeah, so my patient was that he had been type 2 for years on oral, and then had about 6 months of NPH, started getting all different things. Skin test reactive to insulin that I can test them with, and had an IgE to insulin that was positive. And so far, just antihistamine solved its problem.
Okay, I'll try to move pretty quickly, but what I wanted to talk about was this story that's emerged over the last 2 years, really about STAT1 and its role in both severe and relatively subtle immune deficiencies. So I'm going to go through a quick case and then like 4 papers about it that have come out again in the last 2 years. No disclosures. So just skip that. I'm going to give a very brief primer to sort of how we work up the immune system. There are these signs we're supposed to look out for in terms of causes of immune deficiency. And one I think we don't think about a lot, and what's going to be the focus here is this persistent thrush. You probably think about it if you see a neonate with this, but in an adult, probably don't talk about it very much when you're thinking about the immune system. As you recall, we sort of have this paradigm of the 4 main components of the immune system, which is how we're taught as fellows to work up immune deficiencies, and it's great. It obviously catches a lot, but I think the problem that's emerging is this clearly doesn't encompass all immune deficiencies. And we've all seen people who have clear clinical phenotypes of an immune problem that do not have a problem when you work up each of these arms. And I'm going to just blow through this, but these are some of the general labs. We always think about numbers and function within each of those compartments. which again is great, but it does have limitations. So the case that is relevant to the STAT1 story I'm going to tell you, I always think a case is a nice way to help remember things. This is somebody we saw, so it was a 2-year-old or 2-month-old boy who had presented with pneumocystis pneumonia. Had actually a relatively normal immune workup at that time. He had some mildly low CD4 and CD8 count and a slightly high IgM, although that all ended up resolving. He did improve slowly with pretty aggressive treatment. He had a big workup for immune deficiency, all of which came back negative, or for causes of that. He was maintained just because of the severity of his initial presentation on IVIG and Bactrim, and everything grew normally. There were no signs of some sort of syndromic deficiency. But throughout the next several years of his life, he kept getting thrush, and that's what was really the odd thing. And what ended up keying us into what he actually proved to have, which was going to be a STAT1 mutation. We saw him at 3 years. He had a normal immune workup at that time. And then, interestingly, in his family history, and nobody had really picked up on this, so his mom also suffered from recurrent thrush all throughout her life. She had not had the severe childhood infection as he had. And then also his grandmother clearly had an issue with yeast and candida, which gave us a sense that there might be an autosomal dominant genetic disorder going on here. And questions we had is, do we have to care about this thrush? How are we going to treat this kid? And is there a family with PID here? So obviously the issue I ended up having with STAT1, and I'm going to tell you the story again that has come out over the last 2 years. So there were 2 simultaneous publications that came out in mid-2011. One from this group out of England, which was in the New England Journal, and they had a series of patients, I'll go through it in a second, but who had, in this case, quite severe candidal infection. And, you obviously take the picture of your worst-looking patients, but for the New England Journal, that's just an example of one of, or a couple of them. So what they did in this paper to find STAT1 as the likely cause, it was pretty cool. I'm going to talk about what they did and some of the technologies that were clearly helpful, but also the limitations, I think, of this sort of approach. So they had, a fairly good group of patients, 14 from 5 different families, all of whom were suffering from autosomal dominant chronic mucocutaneous candidiasis. They just selected one of those families, which had 3 patients, and they wanted to try to functionally characterize what was going on with their immune cells that would potentially lead to this. So they basically just stimulated their peripheral blood mononuclear cells with Candida, and then measured a variety of things and found that they had low levels of all of these cytokines, especially IL-11 or IL-17 and IL-22, which is known from other work to be relevant to control of fungal infection. They then did something that I think was could have easily failed, but they just picked 100 genes that they believed could be involved in that pathway and said, we're going to sequence these 100 genes and see if we can find it. They then did another thing, which is a little bit of a leap of faith. So they did next-generation sequencing of those 100 genes, which is a technology that's getting better, but it's certainly not perfect, as it has some limitations to it. And they did those 3 patients plus 3 unaffected individuals. You can see the complexity of a question like this. So on average, every patient had over 700 coding variants. So in those 100 genes, there were 700 mutations per patient. Goes to show, how it's going to be tough to do this whole exome sequencing on folks. They excluded things that in public databases are felt to be relatively common or have been seen in otherwise healthy variants, and they were still left with 38 per patient. They then tried to narrow it down further, so they looked for what were heterozygous mutations, and then, by having 3 patients who had it, 3 who did not, they were able to narrow down on really only one gene that was clearly affected in all 3 of the patients and not in the healthy, and it was STAT1. Again, I think there were a lot of limitations here. I think this was a strong enough phenotype and a case where you could collect enough patients that this sort of technological approach ended up working for them. They didn't do a lot to characterize it functionally, but what they did then do is go ahead and sequence, either by next generation or by traditional Sanger sequencing, in the other affected patients, and found similar mutations, i.e. in the same region of the protein within each of those families. So that was, a pretty good study. I actually think this next one, which didn't end up in the New England Journal, it was in Journal of Experimental Medicine, was quite a bit more comprehensive. And this was Casanova's group out of France. He started with a much larger group, so 112 patients, 80 families. He did, instead of this panel of 100 genes that they were interested in, he did whole exome sequencing on 6 patients from 6 different families, which again was difficult because now you're looking at 6 different families, you're going to have potentially 6 different mutations. They didn't actually say how many polymorphisms they found across that, but as you can imagine, it was probably tens of thousands. But they filtered this down, again, using a lot of big public databases, as well as a lot of their own private whole exome sequencing. And with that, they were still left with over 1,100 gene mutations to sift through. And what they did, again, they were cheating here. They obviously knew that the STAT gene was very relevant in immune function. So even though there were a lot of potential gene mutations that could have explained this, they said the only one they thought was relevant was STAT1, realizing this was a group that had already done a fair bit of work with STAT1. All of the mutations, and the other thing I'll point out here is only 4 of those 6 patients had this STAT1 mutation, so 2 of them didn't have it, and they didn't find the cause for it, and you'll see a lot of these patients ended up not finding a cause for the autosomal dominant CMC. But what they ended up doing was sequencing STAT1 in the rest of the, so the other 106 out of the 112 total, and they found in total, I don't think I put the numbers up there properly, but about half of the patients, so 57, I think that's wrong, about half of the patients of that 112 had a STAT1 mutation. A large number of patients, though, again, they did not find a cause for their autosomal dominant CMC. Right, so they sequenced all of them. They only found it in 36 of the patients as an explanation. All of the mutations, they found 12 distinct ones, were all in the same small region of the protein, again, suggesting it to be a cause. And then these guys went ahead, and I'll just show you where the mutations were. So it's all these red mutations, which are in this single domain of the protein, were the 12 that they found in that study. And then this is just how they look in the 3D model of the protein, and basically they're all in this pocket of the protein, and we'll talk about what that meant. But then they went on and did a whole lot more studies to prove that this was actually a functional defect and likely the cause. So first off, they found that those mutations were not in any public databases of healthy individuals or people with other diseases. They used this online tool to try to show that it was a predictive damaging mutation to the protein. They showed, again, that all of these amino acids were in that pocket, which is known to be necessary for dephosphorylation of STAT1. And then they did a fair bit of in vitro work to show that indeed these patients, if you transfect the mutated protein into other cells, it causes delayed dephosphorylation of the protein. Then they showed that these guys, their T cells produce less IL-17 and less IL-22, though they did not show why that's the case. I don't think anybody really knows why that's the case yet. And that's what my last point says, is even though they sort of made these connections, they still haven't been able to spin the whole story as to why these mutations are causing T cells to not respond to Candida and not make those important cytokines. So basically these studies showed that certainly autosomal dominant CMC can be caused by STAT1 mutation, although a lot of patients that's still not the case, so this is not the only protein, which is what I just say there. I think one of the things, again, that interests me about these sorts of studies is these guys generated a huge amount of data with whole exome sequencing and so forth, and most of it just got thrown away. So this is as opposed to how genetic studies used to be done, where every bit of data was pretty much incorporated. Now we're generating huge databases, but we're only targeting down on specific genes of interest. So what else might be contained in that data to explain, the heterogeneity we see in this disease, and all those people who we didn't find an explanation for. The other issue that's been raised a lot with this is, well, again, these studies showed that CMC can be caused by STAT1 mutation. They did not say what do STAT1 mutations actually cause. And what's come out over the subsequent 2 years is that there's a very broad range of phenotype that these guys can have. So while you should think about CMC, you have to think about a variety of other things too. So first off, in that JEM study, they just incidentally noted a fair bit of other stuff in those patients, more than you might expect. In particular, a fair bit of autoimmunity, especially thyroid autoimmunity, squamous cell cancers, and cerebral aneurysms. And people are now trying to figure out how that plays into this disease. So this is Troy's work. He found a variety of patients with this severe IPEX-like syndrome. He found 5 who he thinks it's explained entirely by a STAT1 gain-of-function mutation. And that came out, this was just in June in JACI. And then another group in that same journal, same time, showed 5 patients who were found to have severe disseminated fungal infection. Some of these patients being totally healthy throughout life until one of them, I think at age 17, just developed severe systemic fungal infection. And again, speaking to the range of phenotype here. And then in our family, I think it was interesting, again, most of these patients that are described in these papers are pretty severe. This 3-year-old boy definitely had a pretty severe presentation, but his mom and grandmother had quite mild disease, but had the same mutation. And so it's, what I've learned about this, it's pretty hot topic in the clinical immunology society area. This is probably actually quite common relative to a lot of the immune deficiencies that we think about, that we talk about, like STAT3, hyper-IgE. The fact that we found so many in such a short time, I think, speaks to that. Just briefly, for the sake of completeness, you should know that there are other mutations in STAT1 that were described earlier, although those are all very rare. So again, red are the gain-of-function autosomal dominant mutations that have all come out in the last 2 years. There are also autosomal dominant partial deficiencies, which cause mycobacterial disease. There are autosomal recessive complete STAT1 deficiencies, which are highly lethal, and autosomal recessive partial STAT1 deficiencies. And I just have a quick slide on how those look. So the complete deficiency is a biallelic loss. You basically lose both proteins. 5 or 6 patients have been described. All of them died except one who had a bone marrow transplant. All died very early in life. The partial STAT1 deficiency is much more mild, but it's the same idea. So you have mutated protein, although it still has some function. 5 patients have been described. 4 out of 5 are alive. And then there's an autosomal dominant, which is a dominant loss of function. So it's very much like hyper-IgE syndrome, which is STAT3. You have one mutated protein that causes the other one to not be able to function properly. And there have been 8 kindreds described there, and they have a more mild, they have relatively mild susceptibility to mycobacterial disease, described in O1. Briefly, just to put sense to that, the way STAT1 works, and all the STAT proteins, STAT3 as well, do this. It's a transcription factor. It's a cytoplasmic protein. When it's activated so cytokines work on a receptor within a cell. This is signaled through the JAK proteins, which causes phosphorylation of STAT proteins. They dimerize, which is why if you have one mutated copy, you can still have an autosomal dominant phenotype. They go to the protein, or they go to the nucleus and cause protein DNA transcription and protein translation. So point of this, basically, then there's been a nice review. So, how is it that in one case we can have mycobacterial disease as the main manifestation, and then in another case, Candida? And Casanova's group did a nice review, sort of hypothesizing, but this is supported by some data as to how they think this works. And basically, as you lose function and you have decreased responsiveness to interferon, you sort of fall into this failed interferon pathway, which we've known for a while is related to mycobacterial disease. And with excessive function, so again, where the protein can't dephosphorylate, which is the story I've just told you, in some way it's not worked out, you have dysfunctional IL-17, IL-22, and you're predisposed to fungal infection. So that's pretty much it. I think there's a fair bit that could be talked about here if we have time. Again, points I wanted to make was this clearly plays a role in CMC. It's worth thinking about. It's probably common, but it probably does a lot of other stuff too, and that range of phenotype is still being worked out. So Troy, for example, is looking for this in a whole diversity of patients. We've started to check some patients with immune deficiencies that we can't fully explain at the UW. Then there's also a lot to talk about in terms of the technologies used here to discover this, which I think was cool, but also had a lot of limitations, and there's a lot more that could be done with the data that was generated there.
Your kid, how do you decide which ones need immunoglobulin therapy? You left him on it, and normally immunoglobulin.
Yeah, well, it's a big question, and what we ended up doing with him was we gave him a neoantigen, so we did Hans's favorite test of bacteriophage, and he failed it.
Oh, he did fail.
So, and actually, I presented this at the clinical immunology conference, and it's really the first report of, again, I talked about the range of phenotype, a kid who has really, so you could call him HYPOGAM as well, or CVID-like or something, and that's the sort of scenario where this patient later in life could easily be diagnosed with CVID. Nobody pays attention to the thrush. They just find that they have problems making antibody responses. And, really this gene is. So he's on IVIG, and we're not going to take him off.
It's kind of a mundane question compared to the elegance of the data, but you see women who have chronic vaginal candidiasis and GYN often send them to us thinking, quotes, they're allergic to Candida, just the wrong way of thinking.
Yeah.
Do you think we should screen them for this?
Well, the problem is right now the only test we have is a genetic test, and that's expensive. What we need are better functional tests for this and other clear immune problems. I don't know if all those patients have immune deficiencies. I think David believes that everybody has an immune deficiency of some sort, and I think it's valid.
Oh,
I think everyone who develops thrush, even when he's using inhaled steroids, I think we should test ourselves.
So you think people
Why
who
some
get
people
prone to
develop?
oral steroid-induced thrush have something wrong with them?
Yes,
Well,
I think there's some problem with either in STAT3 or IL-17 secretion, so I don't think that STAT1 is the only problem. You can think about hypomorphic hyper IgE syndrome. Any problem in IL-17 secretion could lead to the same problem. So STAT1 is one of them. But
these two women, his mom and his grandmother, had that. They had mild candidal issues, and they had this mutation. And whether or not it matters for them, probably not. She's already old, but we worry about risk. No, but
We don't care about him.
in him, we're worried because is he at risk for cerebral aneurysms? Is he at risk for severe autoimmunity? We don't know what else. So identifying it in her would be very relevant, not just for her, but for her offspring, because it's going to make you screen the younger people and worry about all these more severe phenotypes. Hers is clearly very mild, and it doesn't really matter for her that she has a mutation.
In that second paper, where there were two families basically where they couldn't find a STAT1 problem, I'm assuming they talked about looking at IL-17 and 22 or their receptors, or see if there were mutations there.
They did. They said very little about it in the paper, and it was more than just two families. So it was actually more than half of the families once they sequenced everybody. They probably did. My guess is they're still working on those patients and looking through their whole exome sequencing, but they didn't find anything to explain those patients.
I guess a deeper question is also the relationship with humoral immunity. You've got an innate pathway that's failing here.
Right.
the kid who's got normal quantitative immune globulins, but somehow is fixed with an IgG replacement.
Right.
So I'm curious how those are connected.
We don't know. I mean, that's, clearly needs to be figured out. I think if we get around to it, Troy and I might work on it a little bit since we have this case. But, I don't know. And, you could hypothesize maybe this kid has a second mutation.
Right.
It's certainly possible, and we don't know. He certainly doesn't have any of the known second mutations.
Matt, in your gain of function stuff, you have 8 or 9 different chemical mediators that are abnormal. Does the disease present the same with all of them? Or have they looked further to see that some people who have just one, or do they have all 7 of these things going wrong?
Which 7 are you talking about?
In your gain of function at the end of your curved arrow, you have interferon gamma, interferon alpha beta,
So
then you have IL-27.
The only thing that I know has been consistently demonstrated in all of these patients who have been checked is that they have poor production of IL-17 and IL-22 on stimulation of their T cells. Many of them also have relatively low Th17 counts if you do it by flow. There probably has been some work. I didn't look at the studies, but I think a lot of these thrown in here are sort of hypothetical, and some of them, from known pathways, and some of them have been really demonstrated, mostly the IL-22 and 17.
And I guess in another practical way, if you have people that don't do well on antifungal therapy, do you try to treat them with immunoglobulins? I mean, that's obviously not anything we would have ever tried on our own.
Yeah, nobody's tried it, or at least nobody's published it. So I think it's a question that needs to be addressed in the future.
There's another case, several case reports actually, that people with gain of function were treated with GM-CSF. And I think there are 2 families, and one of them significantly improved when they were started on GM-CSF, and they showed that the level of IL-17 was increased, and IL-22 as well was increased. And once they stopped GM-CSF again, they developed CMC. So with severe cases, you can think about GM-CSF.
And there's certainly talk out there about transplanting. That's a whole other discussion. But again, it's a big range. There are those who are totally mild, those who are more severe, and it's probably worth identifying mostly because of its clear family history and family disposition.
Bill Anderson typed in a question. I think it's can, but maybe meant, can you demonstrate lack of response to gamma interferon to detect the STAT1 pathway defect?
I think David's doing some of this work, and Troy has a panel to look at the interferon pathway, These patients do not show up on it. So if you just do an interferon gamma stimulation, you will not see it. And I think part of the reason for that is this is a gain of function, so these guys phosphorylate STAT1 just fine. They don't dephosphorylate it. So there is no functional screen for this right now. And I think that's a problem with a lot of these new immunodeficiencies we're finding is we just have a gene, we don't have a functional screen.
Can you replace the STAT1 in the people that are low, or the animals, and show that they get better, and put an antibody against it in the animals that are too high and show that they get better?
I don't know that has been tried, but I'm not sure.
Or can you give them IL-17?
Yeah, can you give them IL-17?
It certainly has not been tried in humans. The mouse models of this, I'm sure there are STAT1, knockouts. I don't know that they've made STAT1 gain of function mice. But all good thoughts. This is a rapid area of study.
Let's save a few minutes for Aaron's presentation. But make sure you speak up, because I've been told before people can't hear you on the outside.
No, that's me.
Speak up, because people have told me after the fact they don't hear you.
That's hard.
Thank you.
You guys can look like your accelerators and gauges and then screen
Down the caffeine.
Yeah, so make sure you spin up is actually offline.
Low molecular weight wonders.
All right, I'm Erin. Nice to meet everybody. No disclosures. And the article that I focused on was in regards to transient hypogammaglobulin of infancy, based on one of the patients we had in immunology clinic. And what I found was that discussion on the phenotypic parameters which could predict the time to normalization in these infants. And notice the irony of the name of the author. All right, so this is a case presentation of our patient. Basically, it was a 12-month-old female who was suffering from reflux and constipation. She underwent food allergy evaluation, including immunoglobulin measurements, and was found to be hypogam. Her initial IgG, and this was at 12 months, was 161, IgA was 8, and IgM was 33. They did check a response to her tetanus antibody, and that was found to be normal. But due to the concern for hypogammaglobulinemia, they referred the patient to Immunology clinic, and this was the GI department at Seattle Children's. On further discussion with the patients, they had not noticed any recurrent infections in the patient, except for one episode of otitis media. Again, she was stay at home with mom, no exposure to other children, no daycares, etc. And then the question became, this family was from China. They were wanting to take their daughter back to meet the grandparents, and they were concerned because no one would give the live vaccines, and they weren't sure if they could postpone their trip to China and what they should do about this. And so that led to my question. But basically, just a little background on transient hypogammaglobulinemia. Is basically hypogammaglobulinemia without any impaired specific antibody responses. So just like in our patient, her tetanus response was normal. It's defined as a transient delay of reaching normal levels of IgG and IgA. However, this is complicating the diagnosis because really you can only make the diagnosis then in retrospect after these patients normalize their levels. All right, so the goal of this study was to define parameters that are present at the time of diagnosis that may help to predict time to resolution of hypogammaglobulinemia and then may help guide treatment options for these patients. So basically 100 infants were identified, and I believe this was from 1978 to 2011. And reasons immunoglobulins were initially obtained were mostly for upper respiratory tract infections, so kids with recurrent otitis media, sinusitis, etc. Occasionally there were lower respiratory tract infections as well, pneumonias, bronchitis. Failure to thrive or GI complaints were the third most common, which would fit with our patient's presentation. Skin infection, recurrent fevers, and some other abnormalities. There were less common. The inclusion criteria that they used for their subjects was basically that the level of any immunoglobulin class was below the 5th percentile for the age-adjusted mean. Their age was younger than 24 months at the time of diagnosis. They were born at term because premature infants are known to have lower levels anyway, so they felt that that would skew the results. There's evidence of protective diphtheria and tetanus titers following immunization, and there was no other known primary immune deficiency. These were some of the statistics. They used the published normal values that were defined by Buckley et al. And basically, they used the lower limit of normal in values less than 2 standard deviations below the mean. Subjects were grouped by the sum of the initial IgG, IgA, and IgM, and then this was repeated based on the IgG levels alone. So at the bottom here, just basically the math formula they used. So they added up the levels in the patient or the subject, and then divided it by the lower limit of normal for all of these levels as well, and used it as a percentile, and then split it up into quartiles. And so basically you can see they did the first, second, third, and fourth. It was nice that they had 25 patients for each. And then the range of immunoglobulin levels as compared to normal is there, and that's how they defined each of the quartiles. You can see that the percent male was quite predominant, and that's just based on the fact that it's 2 to 1 male to female in general for transient hypogammaglobulinemia of infancy. So basically, if we look at my patient and we take her initial levels and we add them up, total of 202, and then the lower limit of normal based on the Buckley paper for the age of 12 months was 438, which is the 46th percentile, so she's in the first quartile. We can use that to help guide our management based on this paper throughout the rest of the presentation.
Erin, I got a quick question.
Mm-hmm.
Have they studied their plasma cells to see what's wrong with them, why they're not producing?
There are other papers that looked at that, and they haven't really found anything conclusive. Some think that it's more of cytokine disturbance, where it's more cellular immunity versus innate, or humoral. Others are looking at Treg levels to see. They found in patients with PHI that their Treg numbers are higher than normal, but they're not sure why. And so they're looking into it. They haven't found a lot in regards to the actual B cells.
But they eventually become normal.
Yeah.
Everything's fine.
Yeah. It's just the problem of you can't technically diagnose them until it normalizes, so everyone looks into what could be affecting it until it normalizes, and then after it's normal, like, looks good.
Since we've interrupted you, why the GI presentation? We don't usually think about that as a presentation for an immunoglobulin test.
No, so I think that comes down to more people testing for celiac and some of those issues as you check the IgA with that now, and if that's low, then people proceed in getting all the immunoglobulins. And then I've seen a lot of referrals for hypogam or low IgA, et cetera, based on these things.
People falling into this for the wrong reason?
It could be. And it could be one of those things that because of it, more patients are being diagnosed with this, just because we're finding it more often now because people are checking for other reasons.
Although I'm sure this is not why they're checking it, but a lot of primary hypogam diseases will present with GI.
Yes.
Right.
Yeah.
Primary or the only manifestation.
Yep.
Why is that the clinical manifestation?
That's a long story, but there are multiple reasons why you have both small bowel and large bowel pathology. So CVID, a classic presentation, is just malabsorption and GI.
Not to beleaguer it anymore, but with Gary's question, have they looked at are there normal numbers of plasma cells or normal numbers of B cells? There are. Okay.
They're just for some reason.
So there's not working.
And just out of curiosity, in the case you presented, why did they do them? Was she failure to thrive, or?
Yeah,
Nope, she was
outside
getting
rate
normally. The story the parents told us was that they were concerned for food allergy, and so they had tested the immunoglobulin levels. It didn't necessarily make that much sense, but
Yeah, sure.
it had been done, and so they ended up in our clinic.
Here's a facetious question, but with healthcare reform, are we going to have the money to keep doing all this?
Quantitative
So-
immunoglobulins.
That's a different journal club. Okay. So these are the results that they found. The median age at diagnosis was 11.3 months. 71% of the subjects were male, which again fit with prior studies. However, there was no statistically significant difference in age of diagnosis or percentage male in the study population was divided into quartiles. So even though there was more males in general, they weren't predominantly in the first quartile or fourth quartile, that type of thing. There was a statistically significant association of quartile of the aggregate immunoglobulin and time to resolution. So basically, if you're in the lowest quartile of immunoglobulin, so that less than 81% of normal, your time to resolution was greater than 5 years, with only about 30% resolving in 3 years. Versus the highest quartile, time to resolution was about 9.9 months after diagnosis, with greater than 70% resolving in 3 years. The middle quartiles were more similar to the highest quartile, but kind of indeterminate at some level.
Aaron, to rudely interrupt again, can you just do specific antigen response and use that? If they respond normally, they're probably okay.
So the problem is that some of the patients do go on to have more problems with recurrent infections, and so the longer it takes for them to develop normal immunoglobulin levels, the higher risk they are at having these recurrent infections.
But no, I'm talking about specific antibody response. Is that some way to show that they have the capability of producing normal immunoglobulin?
Yeah, and that's part of the diagnosis of this, is that they do have a normal antibody response.
Well, that's why I'm asking the question, can you separate them out that way? You know, from somebody that's going to be immunoglobulin deficient and not producing versus somebody that's going to eventually resolve the issue.
Well, apparently not. You said you had a normal response from the get-go.
Well, no, but this is a normal kid. That's what I'm saying. If they have a normal response to specific antigen, then they're probably okay.
Is that true?
Are you referring to the amount of infection? I guess I'm not understanding your question.
Okay, and trying to separate people with a common variable hypogammaglobulinemia don't respond to specific antigen, okay? Whereas these kids do, apparently.
Yes.
So in other words, they can respond appropriately, they just have low amounts.
Yes.
Can you separate out the two, the more severe problem where they aren't going to produce antibody from the transient hypogammaglobulinemia using that test? That's my question.
Yes, and usually if the patient does not have a normal antibody response, then they don't get lumped into the transient hypogammaglobulinemia in the first place, and you start being more concerned about CVID or some other primary immune deficiency. And so they don't really take the abnormal antibody response into consideration in this group.
I'm just saying that maybe they do diagnosis right off the bat and not having to worry and follow them. If they can respond to specific antibody, if they can respond to specific antibody, then maybe you can give them the live vaccines.
That's certainly how you're supposed to try to divide.
Yeah.
The problem is there's a serious gray line there, because you'll have some who respond to one vaccine but not the rest of them.
What if they respond to both, let's say?
Well, if they respond to everything and they have no history of infection, then you don't worry.
Then they're probably okay.
But if they don't respond to pneumococcal vaccine and they've had a couple of infections, and we see a lot of these guys in children, and it's very hard to sort out who's real and who's not.
The other question is, how close to normal do you have to get to be normal? I mean, how much of these immunoglobulins, you define them by being more than 2 standard deviations below, but if you get to 50%, is that enough protection that you don't have to worry about them clinically as opposed to getting to the norm?
And that's the hard part, is it's all variable based on the patient. And so even if you look at people who have CVID and things like that, their IgG level that may be protective in one patient is not in another. And so it's hard to say for sure, well, this patient, because their immunoglobulins are in the lowest quartile, is going to have more infections than the person the highest.
And another thing, you don't define CVID until the age of 4, and you can't define CVID without infections.
Right.
So if they don't have infections, so basically you don't care about the level at this age.
Right.
The main conclusion from this morning, unfortunately, is even for us old people, we're going to have to know something about immunodeficiency.
And the importance of recognizing this is that these individuals, when followed over long periods of time, are at higher risk of autoimmune disease. They develop sometimes other primary immune deficiencies or also IgA selective deficiency. And so that's the importance of just trying to figure out which ones to follow long term, et cetera. How fast do they normalize? How closely do you monitor? Some of those types of things. And so that's why this article is looking at trying to predict, some of those factors so you could basically stratify your patients. Anyway, this is just a graph depicting the time to resolution based on the quartile. So you can see in the 3rd and 4th quartile here at the top, most resolved by age of 5 years. Whereas if you're in the 1st quartile, you're a little bit slower to get back up towards normal. And even then, at 5 years, you still had quite a bit who weren't there yet. And then also they looked at the IgG level alone as a prognostic factor and basically found very similar results. So if you're in the 1st quartile, it takes you a very long time to recover to normal levels versus the 4th quartile. And again, the same gender skew on that. So this is just another graph depicting it based on just the IgG alone. So basically, in regards to the gender difference, they noticed that male infants resolved more quickly than females, and this has also been replicated in prior studies. The median time to resolution in males was about 13 months, with 73% resolving in 3 years, where the median time to resolution in females was greater than 5 years, with 32% resolving in 3 years. So if we take this and we look at my patient who's female, who is in the 1st quartile, she basically has about 30% chance of getting to normal at the end of 5 years. So that's a long time to wait to go to China. And then, again, kind of already discussed, but basically this is the importance of this, is that although the majority of patients have resolution, there was a subset past 3 years that continue to have hypogammaglobulinemia. And these individuals with prolonged hypogammaglobulinemia have an increased risk of recurrent infections and also autoimmune diseases. And so if we have the ability to differentiate or predict the patients that are at risk for this, then this brings up the question of, do we start prophylactic antibiotics sooner Do we consider IVIG, especially in this patient who may be going to China, is unable to get live vaccines? Do we consider IVIG for her time in China to try to provide some immune support? Those types of questions that can be aided by this formula. So currently, with our patient, we're monitoring her about every 3 months, and that's in the hope that she'll somehow recover more quickly and we can give the patient some comfort on their trip to China. However, she's not really cooperating with us at this point. We're still holding off on our live vaccines until the IgG is closer to the lower limit of normal, and for her age, that's anywhere above 350. We have not given her any prophylactic antibiotics or IVIG because she's not suffering from recurrent infections as of yet. Again, she's also not getting a lot of exposures from other children either, no daycare, no siblings. And based on this current article, she might not normalize until 5 years of age.
So, would you recommend she not have exposure to daycare?
Repeat the question.
So it's recommended that in these individuals, even if they do go to daycare, just, hand washing, etc. And it may be that those individuals who are at daycare or exposed to other germs may be more at risk of infection, and the ones that you would consider prophylactic antibiotics, and maybe IVIG, especially during the kind of fall to winter months where they're at increased risk. But then it comes down to more of an art form and how the patient's doing, those types of things.
Can you tell whether there's a defect in B cell function or T cell function in these kids?
So far they haven't found any particular defect. There have been different theories out there. Some thought maybe the B cells were just more immature, not producing, but they haven't found any specific deficiency. And so basically it's just these low levels of immunoglobulins without any other immune deficiency present that they've been able to target.
They tried putting their B cells with a normal kid's T cells and vice versa?
That I'm not sure.
David, you must have something to say about that question.
No, that's interesting. I'll think about DiGeorge. How many of them have partial DiGeorge syndrome that we don't diagnose?
Well, and the other question is, all of our lab values are based on statistics. Maybe for IVIG we should do plus or minus 3 standard deviations instead of 2. Because if all these people are really normal, they're just slow. Maybe we've cut them out arbitrarily by making our cutoff too high, and that they're really normal kids that are just at the bottom.
And that's what they haven't figured out yet either, why there's such a gender difference and why there's more males that have hypogammaglobulinemia, but they tend to resolve faster. They haven't figured out why. So.
More questions than answers.
All right, well, thank you. Very good. Next week, I don't know what we have next week. Anybody else?
Hypersensitivity. Hey, before everybody leaves, does anybody have any really good treatments for itching and people that just can't have any things? Any luck with potions or salves or any of that stuff? Because I got a patient who is wiped out with antihistamine, itching like crazy.